All right, back to proteins. We talked about amino acids, and amino acids are held together by peptide bonds, and proteins are long chains of amino acids. And now we're at the functions. Now, as I have been telling you, proteins are amazing and they're kind of complicated because of that. So you need to pay close attention. Otherwise, the notes will confuse you. Or will confuse you even more than they might otherwise. So right here, oops, forgot to set the pen again, always. So if you notice in the notes, right here it says functions. And there's 2 points, A and B. B is the actual functions. A, the function depends on organization. So point, let's see, point A, point A then is organization. And why is organization under function? Because the function depends on organization. And then B in the outline are, in a general sense, the different kinds of functions. So today we're going to talk about organization. The function of a protein depends on the protein's organization. The organization is critically important. It is so important that if you change the arrangement, you destroy the protein's ability to function. And when a protein's ability to function is changed, we say the protein has been denatured. So when I was a kid in junior high, and this definitely sounds like a junior high kind of thing, some days for hot lunch, they would have pickles, dill pickles. Dill pickles, if you don't know, are in vinegar, which, if you don't know, is an acid. And so some of the junior high kids would occasionally take their dill pickle juice out of the little cup, little paper cup that the dill pickles were in, and they would pour it into their little half pint or whatever it was carton of milk, and the milk would instantly curdle. It would instantly look like rotten, except it wasn't rotten. Why did that happen? Because the acidity of the pickle juice denatured the proteins in the milk, and the proteins in the milk became clumps instead of dissolved like they're supposed to be. Or maybe you know that if you get grass stains or blood stains on your clothes, you are supposed to wash them in cold water. Why is that? It's because hot water denatures the protein in the blood or in the grass. And once it's denatured, it is no longer soluble in water, so it becomes permanent. It sets the stain, as we say. So the function of protein is absolutely critical. It depends absolutely on its organization. And if the organization changed, the protein won't work. It's said to be denatured. So we have 4 levels of organization in proteins. First 2 of these words you're probably familiar with. You're probably familiar with the term primary, and you're more than likely familiar with the term secondary, but you're maybe not so familiar with tertiary and quaternary. Tertiary is 3, like 3rd degree. Quaternary is 4th degree. I don't suppose that it's critically important that you know the terms primary, secondary, tertiary, and quaternary. At least not for this class. If you do much in chemistry, organic chemistry, and biological chemistry, then you should know the terms. But 1st degree, 2nd degree, 3rd degree, 4th degree, first, 2nd, 3rd, 4th levels, that's easy enough to understand. So we're going to have to do some note filling in here. The primary structure is the specific sequence or order of amino acids. And this specific sequence is determined by the protein's gene. That is actually what genes do. Genes determine the sequence of amino acids in a protein. I mean, it's just that simple. Why do you have blue eyes if you have blue eyes? Whatever color eye you have, why do you have that color eye? Because the genes for eye color determine the sequence, the order of the amino acids in the eye color protein, and the particular combination of the particular combination of amino acids determined by the genes you got make the color of your eyes. Why do you have the level of pigmentation in your skin, the darkness or lightness of skin, or the shade, be it more of a reddish shade or a brownish shade? It's because of the specific order of amino acids determined by the particular gene for a couple of genes, amount of melanin and kind of melanin. So why do you have whatever you have? It's because of the specific order of amino acids in the proteins as determined by the genes you got. So this is quite important. I mean, all of these are important. Primary structure, it all depends on primary structure. In other words, the specific order of amino acids. There is a genetic condition that you may or may not be aware of called sickle cell anemia. It's called sickle cell because it makes the blood cells, normal blood cells are supposed to be shaped something like this, with a little shallow spot in the center, not a hole, but a dent. So normal blood cells are supposed to be round, even rounder than I drew it. In sickle cell anemia, instead of being shaped round, they kind of tend to be shaped kind of in this bent shape, like a sickle. A sickle is a curved knife that is used to cut grass or whatever. It seems like one of the communist countries, China or Russia, might have a sickle on its flag. Maybe, I don't remember for sure. Anyway, this is what carries oxygen. This is good. You can carry oxygen just fine. 2 men in a canoe. This is not good. You can't carry it so well. 2 men in a capsized canoe. So I don't know why men. Maybe they're girls. I don't know. Maybe they're women. I don't know. But the difference between normal and sickle cell anemia is one mistake in the gene, so there is one amino acid out of place in the protein. So primary or the specific order of amino acids is very important. Sometimes you can have a mistake and it doesn't cause a problem, but in certain circumstances, just one wrong amino acid can mess the whole protein up. All right, so we get this long chain of amino acids in the primary structure. As I think I told you yesterday or the day before, hemoglobin, the stuff in the red blood cells, is 4 chains of about 200 each. There are 2 alpha chains and 2 beta chains. Alpha, in case you don't know, is the first letter of the Greek alphabet, and beta is the second letter of the Greek alphabet. And this is just basically saying, that there was chain A and chain B, 2 copies of chain A, 2 copies of chain B, 2 alphas, 2 betas. Okay, so once we get our long chain, 200 give or take of amino acids, specific amino acids, not just any amino acids, but specific amino acids. Once we get our long chain of these, then we do the secondary structure. The secondary structure is taking this chain from the primary structure, and it is the coiling. Those are called alpha. There's the sign for alpha. That's not a D. It's like a small A with a little tail on the top. Alpha helix, or folding back and forth. I'm going to not say back and forth because I'm going to run out of room for writing maybe. And that would be the beta. Now that looks like a capital B with a tail coming off the bottom. Beta pleated sheets. The coiling, alpha helix, oops, or not of. The coiling, alpha helix, or folding beta pleated sheets of the chain from number 1, primary structure. These are held together by hydrogen, H for hydrogen, bonds. Bonds, James bonds, not bombs. Kaboom! So secondary structure, coiling, alpha helix, or folding the beta pleated sheets of the chain from the primary structure, held together by hydrogen bonds. Now we can give you a little bit of an idea of this. I had a window open, where'd it go? Oh, it's right here. I had it open and nice and big and tall so that you could see it. Oh, there it is. So here is what we mean by coiling. Now, you don't have to know this, but remember you do have to know this, but not this exact picture. So there's our amino acid, nitrogen, carbon with the R group, and the carboxylic acid attached to the next amino acid. And in the alpha helix, this chain just makes this looping coil back around and round and round for a certain distance. It doesn't go forever, just part of it does. This is trying to show the same thing except using ball and sticks to show you where the different nitrogen and carbon and hydrogen and R groups are. And these dotted lines, or these look like red dashes here, are showing you the hydrogen bonds that hook this together. So some of that long chain, which there might be a picture of up higher if I go, yes. So here is what you start with. Here is the primary structure, and each different color on this represents a different amino acid. So see, they're held together by peptide bonds and the different amino acids. There's the N and the C and the double bonded O and the R, and there's no OH because they have been, it was removed in the peptide bond. But we get this primary structure, and it's not random. It's a specific order determined by the genes. And then once we do that, some of the area coils and is held together by hydrogen bonds. Some of these loop back and forth. They come and they loop back, and then they loop. They're only showing you a little bit. It would be like they come and they loop back, and then they loop back, and then they loop back, and they make a sheet. But the sheet goes down and up and down and up. So it's a pleated sheet, you know, pleated like a pleated skirt or like a curtain or whatever. And still we have these dotted lines. They're held together by hydrogen bonds. So the specific order, and then they coil into the alpha helix or fold into the beta pleated sheet. These are, remember, held together by hydrogen bonds. And then the tertiary structure, now I'm just changing color on these because it's hard to write on the screen and stay in the allotted space, and I want you to not get mixed up of which line is which. The tertiary structure is the alpha helix, alpha helixes, the actual word is helices, but alpha helix and beta pleated sheets fold back and forth over each other. And these are held together by disulfide bonds and some other stuff. Disulfide bonds, etc. So if you don't have it written down, I'll be back in a minute. So if we look down a little bit, here we have tertiary structure. Here you can see an alpha helix. Here you can see another alpha helix. Here's a beta pleated sheet. And notice how this alpha helix folds back over top. So this is folded over top of the sheets. And then the second alpha helix folds a little more. And right there you have a sulfur and a sulfur. There's two of them. That would be disulfide. So that's what holds them together. Like I said, there are other things that hold them together. There are hydrophobic interactions shown as the orange, hydrophilic interactions shown as the green, and then there are acidic and basic amino acids. So there's actually several things that can hold this tertiary, excuse me, tertiary structure together. But the only one I'm asking you to know right now is disulfide bonds. And then finally, we've got the quaternary structure, or fourth degree. And the quaternary structure is two or more folded chains with folded in the secondary and tertiary structure combined. So I'll be back in a minute if you don't have that written down. So if we go down a little further here, this is hemoglobin. More or less, sort of. So you see we've got in our quadrant, we've got the yellow dotted line, the upper left. There's one of the chains of one of the polypeptides of hemoglobin. And then the green dotted lines on the axis, the second of the chains of hemoglobin. And then the blue dotted lines for the third folded and pleated chain of hemoglobin. And the red dotted lines for the fourth folded and pleated chain of hemoglobin. So it takes those four chains, 2 alpha chains and 2 beta chains. Remember, okay, so we've got alpha helix and alpha chain. It's just like saying A and B. We've got chain A and chain B, and we've got A, which is the coiling, and B, which is the pleats. Just a way to tell them apart. The A and the B, or the alpha and beta, don't mean anything special other than this one and that one. But when we put those together, 2 or more folded polypeptide chains combined, and we get the final functional protein. So the primary structure, specific order of amino acids, the secondary structure, coiling into an alpha helix or folding into a beta pleated sheet, and it's sections of the chain. It's not all of the chain that does any one of those. That's from the primary structure. They are held together by hydrogen bonds. Tertiary structure, the alpha helix and beta pleated sheets fold back and forth over each other. These are held together by disulfide bonds and so on. And then we take 2 or more of those chains and put them together. And I say again, it's not just any old 2 chains. Are the chains that are determined by the genetic code to belong together. So all of this is very, very, very specific. You know that protein that runs along a protein rail? It has one particular set of genes making one particular arrangement of coils and folds and folding back and forth over each other. But then that generator protein I told you about has a completely different primary, secondary, and tertiary and quaternary structure, different order, different folds, different combinations. So each particular protein has its own specific arrangement of these things. All right. That's all I'm going to cover today. You have the PFG that needs to be done. You have a quiz to take. Other than that, you're free to go. It's just a little bit less than half an hour, but not really a time to start the next one, next point. And have a good day.